Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. In addition, I'll tie in some of the testing strategies that I and other students have used on the MCAT. This podcast will cover renal physiology, and this is one of the most important topics to understand regarding the excretory system that's going to show up on the MCAT. First, I'm going to go over a little bit of anatomy, just what's necessary so that you can understand the physiology. And then I'll go over different functions of the kidneys and the mechanisms that the kidney uses in order to complete these functions. And then I'll talk about 3 hormones that affect the kidneys, more specifically affect the nephron, which is the functional unit of the kidney. And then lastly, I'll briefly mention a few measurements of renal function, such as glomerular filtration rate. And I think this is a good podcast for really anyone who wants to understand the kidneys, which are pretty interesting organs. And so, if you have a friend maybe that's interested in understanding how the kidney works, maybe they work for a dialysis company or maybe you have a friend who's interested in healthcare policy and understands that there is a lot of people in the US that suffer from these kidney-related diseases. It's estimated that 1 in 3 people have chronic kidney disease. And obviously that's a lot of healthcare to pay for. And a lot of these patients are actually covered by Medicaid. And so the government's paying for a large fraction of this healthcare. So if you're interested in healthcare policy, you should be interested in the kidneys as well. So pretty interesting, share this podcast with them if they're interested. Thanks for listening as always, and I hope you get something good out of this podcast. All right, as I said in the intro, the first thing I want to go over here is some basic kidney anatomy. So obviously, if you're in front of a computer or have your phone or, just listening to this really trying to study, it's best to have an image of a cross-section of a kidney to kind of follow along with what I'm talking about. But if not, no big deal. Maybe look up a picture when this podcast is over. All right, so in general, you have 2 kidneys. They're bean-shaped and they are found in vertebrae only. And you have 1 kidney on each side of the spine. And the parenchyma of the kidney, which are the functional cells of the kidney, are broken up into 2 different categories. That is the outer renal cortex. And you can kind of think of this a little bit like a cerebral cortex, right? That's the outer layer. The cerebral cortex is the outer layer of the brain, as is the renal cortex, outer layer. And then you have an inner layer of cells called the renal medulla. And there is a little bit of difference between these different layers. The renal medulla itself is a little bit more salty or has a higher osmolarity. There's more solutes. And this plays an important role in the loop of Henle. And I'll get into that and discuss that in detail when I talk about the nephron. And further, the renal cortex surrounds bits of the medulla called the renal pyramids. And they're kind of these cone-shaped structures that reside within the kidney, which to me looks a little bit like if you took an orange and just sliced it in half, and then you see the peel around, and then you have these almost cone-shaped bits of orange, right? If you were to cut it cross-sectionally, you'd see what I'm talking about. But you can kind of imagine it like this. So these renal pyramids are bits of medulla that are within the kidney. And between these renal pyramids are renal columns. And these are just projections of the renal cortex. And you definitely wouldn't get a question about these on the MCAT, but it's still good to know in order to have an understanding of the renal system. And as I said earlier, the nephron is the functional unit of the kidney. It is to the kidney as the mitochondria is to the cell. It's a powerhouse. And these nephrons span from the renal cortex all the way into the medulla. And blood is supplied to the kidneys via renal arteries. So these arteries come in, they come towards the kidneys, supplying blood to them, and they split a few times into smaller and smaller arteries, eventually producing afferent arterioles. And that's what enters into the glomerulus, and that's where filtration occurs, and I'll get to that in a bit. But just know, from these renal arteries, you go into the afferent arterioles, through the glomerulus, and then out into the efferent arterioles as you leave that glomerulus. Before I go any further, I want to note that the word glomerulus, it is the word equivalent of pouring honey. It sounds like molasses. It's the word version of molasses. Just glomerulus. In my opinion, it's kind of a bit of a gross sounding word. All right, so the last thing in terms of anatomy I want to talk about is the anatomy of the nephron itself. The nephron itself is made up of 5 really important different locations of interest in which different things happen. And that's obviously very vague, and I'll get more into what happens at these different places when I talk about the physiology of the nephron and of the kidney. But first, I want to just introduce to you these different locations within the nephron. And I'm going to present these in the order that an excreted molecule would see. So for instance, let's take asparagic acid. This is the component of asparagus that makes your pee smell funny when you eat it. And so you're a little molecule of asparagic acid. You're in the blood. You make your way down the afferent arteriole. And the first part of the nephron you see is called the renal corpuscle. And this contains Bowmans Capsule and the glomerulus. And then once you make your way actually into the nephron there, you get filtered through the Bowmans Capsule, and you make your way actually into this tubular structure that is the nephron. And go Google an image of this if you really want to see it. It's this long kind of twisted tubular structure. And the first part of the nephron, after you make your way through Bowmans Capsule, is called the proximal convoluted tubule. So you make it into there, you travel down, and you go down the first part of this loop and then up the second part of this loop. And this loop is called the loop of Henle, which is the third structure within the nephron to know. It has a descending limb going down and an ascending limb. And there's different things going on at each of these different limbs. All that's important to know. Then once you get past the ascending loop of Henle, you make it into the distal convoluted tubule. And the proximal and distal convoluted tubule are called convoluted because it kind of folds over itself. It's basically a big squiggle folding over itself. Then the very last place, or the 5th location to know within the nephron, is called the collecting duct. And this collecting duct is actually the last step in either concentrating or diluting the urine. And I'll get into that when I talk about physiology. And after the collecting duct, the material to be excreted, again, this contains the asparagic acid that we talked about earlier. This, along with mostly water, travels through the collecting duct into the ureter, which collects urine from all the different collecting ducts, kind of like the mother of all collecting ducts. And then that ureter travels into the bladder, and your urine is stored there until you urinate it out. So that's really all you got to know in terms of kidney anatomy. There are these bean-shaped organs. On the outside, you have a renal cortex, and on the inside, you have a renal medulla. And these renal medulla are these pyramid-shaped bits, cone-shaped bits that are surrounded by the cortex. And the cortex projects down actually into the kidneys through these things called renal columns. And then the nephron is broken down into 5 distinctive parts. That is the renal corpuscle, the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and then the collecting duct. So that's really all you got to know. Again, the MCAT really doesn't test much of anatomy. Instead, they focus a lot more on physiology. So let's get into renal physiology. So as I said previously, the nephron is the functional unit of the kidney. This is what you're going to see on the MCAT, the nephron. You're going to hear a lot about this on the MCAT if you see a question. And this is not to be confused with the very similar Zac Efron, which, if I understand correctly, actually stimulates a whole different organ system. No, just kidding. All jokes aside, though, kind of interesting. I don't know if any of you have seen pictures of Zac Efron lately, but he dyed his hair like completely like this silver blonde color and has like this dark black beard, and it looks kind of cool, honestly. I'm really thinking of adopting that look just because I think it looks kind of cool. But anyways, back to the nephron and the kidneys. So the kidneys have these 6 main functions, all that kind of help the kidney maintain the body's homeostasis. So the first function of the kidney is to regulate plasma fluid volume. And this helps regulate blood pressure, right? So if your plasma volume is very high, then your heart has to pump more blood, and it's got to put more work in to do that, and your blood pressure is higher. But if you have a low plasma volume, then your heart has to do less work because it's pumping less blood, and therefore your blood pressure is lower. The second function of the kidney is to regulate the osmolarity in your blood. So that's just the amount of solutes that are dissolved in your blood. The third function of the kidney is to regulate ion concentrations. And so this is, regulation of sodium, calcium, all these really important ions that are important for other physiological functions. Think about how many bodily processes are dependent on some sort of ion. So, for instance, the firing of a neuron requires sodium gradient, requires potassium. You have a sodium-potassium ATPase, which relies on those two ions to function. You need calcium for muscle contraction. So it's very important then to maintain the concentration of these different ions in the blood and in the body. The fourth main function of the kidney is in regulation of blood pH. Very important. And the fifth function of the kidney is in excreting wastes and toxins. And then the sixth function of the kidney is in the production and release of a few different hormones. Some of the main ones are erythropoietin, which stimulates red blood cell synthesis. Next, there is calcitriol, the active form of vitamin D that promotes intestinal absorption of calcium in the renal reabsorption of phosphate. Then the last one is renin, which is an enzyme that regulates angiotensin and aldosterone levels and plays a role in blood pressure regulation. All right, so that is what the kidney does. I want to get into how the kidney does these things. So many of these functions can be tied back to how the nephron functions. And this is really the most important information to know for the MCAT. So again, the nephron, as I've said a million times, is the functional unit of the kidney. And it operates through 4 different processes. That is filtration, reabsorption, secretion, and excretion. These processes occur throughout the nephron at different points. And so what I want to do is I want to go through the nephron, present each of these stages of the nephron that I previously mentioned when I was talking about anatomy, and then talk about the 4 processes that occur at that step or that don't occur at that step. And this will help you kind of understand what's going on at the nephron at each point. So let's rewind just a little bit. I am a molecule traveling in the bloodstream, and I'm making my way down to the kidneys. I'm close to the kidneys. I go down the renal arteries, make my way into the afferent arteriole, and make my way to the renal corpuscle. And again, the renal corpuscle contains 2 different parts, which are the Bowmans Capsule and the glomerulus. So you've made your way down to this renal corpuscle, and the first thing you encounter is the glomerulus. The glomerulus is a small tangle of capillaries. It's almost like this interweaved kind of mess of capillaries. And you can kind of imagine this glomerulus as having a round shape. And around this glomerulus is what's known as Bowmans Capsule. And so Bowmans Capsule is essentially the filter for the renal corpuscle. And it's made up of these cells that have a little teeny gap between them. And then what this allows is very small molecules to flow through those slits and get filtered into the nephron. And so small molecules like water, ions, glucose sometimes shouldn't be, but sometimes gets into the nephron, whereas big proteins and other large molecules are rejected from that filter because they can't fit through these filtering slits that are created by the gap between cells that make up Bowmans Capsule. And so you might be thinking, okay, cool, the Bowmans Capsule filters, I get that, but then what is the point of the glomerulus? What does this do? So the glomerulus, again, is this small tangle of capillaries, kind of looks like a ball of yarn or something. And what's interesting about the glomerulus is that the cells that make up the glomerulus have these things called fenestra. And so fenestra are essentially these holes in the cells. And this allows a bunch of molecules to get through that can then make it to those filtering slits in Bowmans Capsule and then be filtered. So essentially, the role of the glomerulus is to be leaky, let these molecules get out and be filtered. And I should note here that the glomerulus actually does filter some things too, so it doesn't let white blood cells or red blood cells get through into the nephron. So, you probably shouldn't see blood in your urine. Obviously, for different reasons, you might have a UTI or something like that and you see blood, but that's obviously not normal. And so the reason that you don't just pee blood all the time is because the glomerulus actually filters out the blood. So these red blood cells are kept in the capillaries because these fenestra are not big enough to allow the passage of these red blood cells through to Bowmans Capsule and then into the nephron. So as I'm sure you understand by now, the main function of the renal corpuscle is to filter the blood. So this is the one and only stage in which filtration occurs. And again, filtration is one of the 4 processes by which the nephron operates. So if you think about the renal corpuscle in terms of filtration in general, you'll be thinking, how are these molecules forced through? When you filter something, you need some kind of force or some kind of pressure to push that fluid through the filter. So the answer to that question is that higher pressure within the blood actually forces that blood through the renal corpuscle and into the nephron. And so you can kind of think of this as like a syringe filter. I don't know if any of you have worked in a lab. I'm assuming that either a lot of you have worked in a lab or will work in a lab. If you've ever used a syringe filter, you know that essentially it is like a syringe that you use for injections, but you screw on a filter on top. And then you have to apply pressure to that plunger then in order to force liquid through the syringe. So in the same way, plasma from the blood is forced through the renal corpuscle via higher pressure. So the blood pressure is a lot higher than the pressure within the nephron. This difference is the driver for filtration. Okay, cool. So a brief recap. Filtration occurs at the renal corpuscle. This is the only point where filtration occurs. And the glomerulus is made up of these cells that have fenestrations or are fenestrated. And these are just holes that kind of are permeable to large molecules and small molecules. However, red blood cells, white blood cells aren't able to get through those fenestra. And then you have a Bowmans capsule, which is also another layer of filtering. You have these cells with slits in between them that essentially block larger molecules from getting into the nephron. And they are able to permit small molecules like water, ions, sometimes glucose into the nephron. All right, so let's assume you are the asparagic acid and you've made it through the filter. Good job, you're in the nephron. The first part of the nephron you encounter is the proximal convoluted tubule. And it's important to note that the proximal convoluted tubule runs in parallel to a set of capillaries called the peritubular capillaries. These are essentially the capillaries that are after the efferent arteriole. And again, the efferent arteriole is the arteriole leaving the glomerulus. So you go from glomerulus to efferent arteriole into the peritubular capillaries. All right, so there are 2 huge processes that go on within the convoluted proximal tubule. That is reabsorption and secretion. Reabsorption is the movement of solutes from the nephron into the blood. So that is that species that made it through the filtration and are now in the nephron are just reabsorbed back into the blood. Whereas secretion is the movement of species from the peritubular capillaries into the nephron. So they're just opposite processes. Just remember, reabsorption is reabsorbing from the nephron back into the blood for use in the body. And it's important to note that in reabsorption and secretion, the movement of these different compounds from the blood to the nephron or vice versa can happen via a bunch of different mechanisms. So, this can just be diffusion, it can be passive transport, it can be active, it can be secondary active, it can be, every single different kind of transport you could ever imagine. So just keep that in mind as I go through this talk. I'll try to kind of mention which ones are active and talk about different transporters and stuff. Just keep that in the back of your mind. So let's first start by talking about reabsorption that occurs in the proximal convoluted tubule. So again, reabsorption is the movement of solutes from the nephron into the blood. And there are a bunch of different compounds that are reabsorbed in the proximal convoluted tubule. So we have sodium is reabsorbed. Very importantly, glucose and amino acids are reabsorbed. Water's reabsorbed. Potassium's reabsorbed. Urea, phosphate, and citrate are all reabsorbed at the proximal convoluted tubule. Let's get a little bit more into detail about a few of these compounds. So sodium is absorbed via diffusion and also actively via a sodium-potassium ATPase pump. And then glucose and amino acids are reabsorbed using secondary active transport. So these use a sodium concentration gradient that's been built up by the sodium-potassium pump to then pump the glucose and amino acids back into your body. And obviously your body's telling the kidney, what are you doing trying to get rid of this glucose and amino acids? I can use the glucose for energy. I can use the amino acids for different metabolic processes, including making proteins. Please give me those back. I'm going to use those. And so your body reabsorbs them. It's important to note here that in healthy patients, glucose is never secreted in your urine. However, if these transporters that transport the glucose back into your body become saturated, and this is when you have really, really high blood glucose levels, then you start to secrete glucose into your urine because obviously with the saturated transporters, you can't get enough of this glucose back into your body in order for it to not be excreted. And typically this is an indication of diabetes. And it's said that water follows the solutes that are reabsorbed in this stage. And so what do I mean by that? Essentially, you want to maintain the osmolarity in the nephron and outside in the blood. And so water is then driven out of the nephron into the blood to kind of try to maintain that. The next thing that I want to talk about that is reabsorbed is citrate. And so citrate helps us regulate pH. A 2009 paper by Arnson and others said that after entering the cell, reabsorbed citrate is completely metabolized by mitochondrial oxidation to yield bicarbonate, which is used in the body's blood buffering system. All right, so let's talk about the molecules that get secreted. And again, secretion is movement of molecules from the pertubulary capillaries to the nephron. So these are going to get excreted. There are three kind of important compounds that get secreted in the proximal convoluted tubule. That is many drugs, and you don't really need to know which, just know that a lot of different drugs are actually secreted at this stage. An example is penicillin. And then creatine is also secreted at this stage. And then lastly, uric acid is secreted in the proximal convoluted tubule. So the takeaway for the proximal convoluted tubule is that the body reabsorbs all the stuff that it needs that made it through the filtering system, and it secretes drugs to be excreted. All right, so that's the proximal convoluted tubule. Let's go back to the asparagositic acid. You're this little molecule. You made it through the proximal convoluted tubule, and now you go down this loop and then back up. And this is the loop of Henle, and this is the next structure to understand in the nephron. So the loop of Henle is found between the proximal convoluted tubule and the distal convoluted tubule, and it carries filtrate, which is the liquid that is filtered into the nephron, from this proximal tubule, which is in the renal cortex, down into the renal medulla, and then back up to the renal cortex. Hence the loop part of the loop of Henle. The Henle part of loop of Henle comes from the scientist who discovered this loop. His name was Friedrich Gustav Jakob Henle, which is a pretty sweet name. Anyways, the portion of the loop that goes from the cortex into the medulla, which again is the outside of the kidney into the inside, is called the descending limb of the loop. And then the portion of the loop that goes from the medulla back to the outside, which is the cortex, is called the ascending limb of the loop. And so what's the importance of this loop? I.e., what is the difference between the renal cortex and the renal medulla? Let's get into that. The osmolarity of the interstitium of the kidney increases as you go from the renal cortex into the medulla. And so the interstitium is just this fluid-filled space outside the nephron. So you could imagine as you go from the cortex down deep into the medulla, it gets saltier and saltier, more and more solutes floating around. This is really important because the difference in osmolarity between the filtrate inside the nephron and the fluid outside the nephron increases as you go down the nephron. And this is the driving force for diffusion. So this is to say that as you go down the loop of Henle, the driving force for diffusion for a bunch of different molecules increases. So it's like going from a 1995 slug bug to a 2019 Ferrari, right? You just got a bunch of horsepower. Your driving force just increased a ton as you went down this descending limb. So I think the really important thing to understand here is that as you go deeper into the kidney, into the medulla of the kidney, it starts to get saltier and saltier, and this increases the driving force for diffusion of a bunch of different molecules within the nephron.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
So let's first talk about reabsorption when it comes to the descending limb. So again, reabsorption is the movement of molecules from the nephron back into the blood or back into the systematic circulation. So the descending limb is very, very permeable to water, but is not very permeable to ions. So let's think about this in terms of osmolarity, right? As you go down this descending limb, the osmolarity outside is increasing. So to balance that out, you get an efflux of water. That is to say that water is reabsorbed as you move down this descending limb. And if you're struggling with the concept of water moving to balance out osmolarity, think back to biochem of these setups where you have a semi-permeable membrane, and on each side you have a solution with concentration X and concentration Y on the other side. And what happens is you'll have flow of water to equal out the concentrations on both sides. So in that way, you can kind of think of the descending limb of the loop of Henle as kind of a semi-permeable membrane that allows water to flow through, but is not very permeable to ions. The ascending limb of the loop of Henle is also called the diluting limb, and it's kind of the exact opposite of the descending limb. It is impermeable to water, but is very permeable to ions. So at this point, ions are transported out of the nephron. They are reabsorbed. The most important ion that is reabsorbed is sodium. So this happens in two different ways. So sodium is reabsorbed through a transporter known as the sodium-potassium ATPase, which I've talked about before. The other is through a co-transporter called the NKCC2 transporter. And so what happens is you transport a sodium, a potassium, and 2 chloride ions all at the same time. So this is a co-transporter. In addition to these two mechanisms, sodium can also leak through between cells. This is called paracellular transport. And so the same goes for calcium and magnesium. Both those ions can go between cells and be reabsorbed back into the blood. So you kind of have a wide variety of ions being reabsorbed here, right? You have sodium, potassium, chloride, magnesium, and calcium. And again, this ascending limb is also called the diluting limb because you have movement of ions out of the nephron, but you retain all the water. So what's going on? You're just diluting the urine at this stage. All right, so to quickly summarize the loop of Henle, in the descending limb, water is reabsorbed, driven by this osmolarity gradient between the filtrate inside the nephron and this salty fluid surrounding the nephron. And then once you get into the ascending limb, water can no longer be reabsorbed, and now you reabsorb a bunch of ions, and this is also called the diluting limb. And in terms of the loop of Henle, I'd only really worry about knowing the reabsorption that occurs, which is what I just talked about, and really understand how osmolarity of the interstitium plays a role in forcing water out of the nephron for the descending portion of the loop of Henle. So after this filtrate has passed through the loop of Henle, it enters the distal convoluted tubule. And cells of the distal convoluted tubule are very rich in mitochondria, which means that these cells are doing things, right? They need a lot of energy. And so these cells possess the highest density of sodium potassium ATPases along the nephron. So obviously the fact that these cells have a lot of mitochondria and a lot of sodium potassium ATPase pumps is very interconnected because you need a lot of ATP in order to operate these pumps, and obviously mitochondria are good at cooking up ATP. So just know that there is a lot of transport going on in the distal convoluted tubule. So in this section of the nephron, there are a few important ions that are reabsorbed. Sodium is reabsorbed and chloride is reabsorbed. And so the distal convoluted tubule reabsorbs about 10% of the sodium and chloride that get filtered into the nephron, which is a decent fraction. And then calcium and magnesium are also reabsorbed in the distal convoluted tubule. So that's 4 ions to know that are reabsorbed in this section. In terms of secretion, there's a few interesting molecules to know. So again, secretion is movement of a molecule from the blood into the nephron to be secreted. So at this stage, potassium is secreted. According to a review article that I read, it says that many studies indicated that potassium secretion along the distal tubule accounted for most or all of potassium excretion under a variety of conditions. So that's to say that the distal convoluted tubule really controlled the output of potassium in the urine. So that's important to understand, that the way that the distal convoluted tubule controls the secretion of potassium, results in how much is excreted. So if the body has too much potassium, then there'll be a lot of secretion of potassium so that it can all be excreted. If there's not much potassium in the body, well, then the secretion of this potassium into the nephron is not going to occur as much. So that's important to understand. And then the next molecule that is secreted at the distal convoluted tubule is urea. And then the third is hydrogen ions. And this occurs via a sodium hydrogen exchanger, where you get one sodium out and one hydrogen ion into the nephron. And so this is one of the mechanisms by which the kidney maintains blood pH, right? If you have too much hydrogen ion built up in the blood, therefore you have a low pH in the blood, then the kidney can actually excrete this hydrogen ion through secretion in the distal convoluted tubule. All right, so that is the distal convoluted tubule. What's important to know there is you reabsorb sodium and chloride, and then you secrete potassium, and also hydrogen ion, which is important for pH balance. So next, we go into the collecting duct. So this is the final location within the nephron, and it's responsible for the final concentration of the urine. And so it is water permeable, but this permeability to water is actually controlled by a hormone, which is ADH, or antidiuretic hormone. And what antidiuretic hormone does is it actually increases the formation of aquaporins within the collecting duct. And I'll get into this in a sec when I talk about hormones, but just understand that the collecting duct is responsible for the final concentration of urine. And so there are a few different molecules that are reabsorbed in the collecting duct. The first is water, obviously, as I just talked about, and vasopressin, or antidiuretic hormone, plays a big role in this. And interestingly, in times of extreme dehydration, like let's say you're in a desert or something and you're really dehydrated, about 25% of the filtered water will actually be reabsorbed through the collecting duct. So the collecting duct can really either concentrate your urine or really dilute your urine, depending upon your environmental surroundings or, your homeostatic state of being, whatever it may be. The collecting duct has a lot of power in terms of concentrating your urine. You also reabsorb sodium, chloride, bicarbonate, and urea at the collecting duct. And I think it's important here that I talk a little bit more about the regulation of pH by the kidney. So I may have failed to mention that in the proximal tubule, a lot of bicarbonate is reabsorbed. So I think like 85 to 90% of bicarbonate that is filtered into the nephron is reabsorbed in the proximal tubule. The rest is reabsorbed in either the collecting duct or the distal convoluted tubule. And this bicarbonate can go on then to react with hydrogen ion that's, let's say, in excess in the blood and neutralize that hydrogen ion. It's part of the body's blood buffer system. And so through regulating how much bicarbonate is reabsorbed and how much hydrogen ion is secreted, the kidney can therefore help maintain the body's pH. So that's pretty important to understand for the MCAT. All right, back to the collecting duct. So there are 3 molecules that are secreted in the collecting duct that I'm going to talk about. The first is potassium. The second is hydrogen ion. And the third is ammonia. Those are 3 things that are absorbed within the collecting duct. More important to know, though, is how ADH affects the collecting duct, and to know that the collecting duct determines the final concentration of urine. All right, going back to our asparagic acid example, the little molecule has made it all the way through the distal convoluted tubule into the proximal convoluted tubule, and now it goes into the ureter and makes its way into the bladder, where it will be excreted out the urethra. And it's kind of interesting that for some people, you'll smell this right away. I can eat a single stalk of asparagus and literally 5 minutes later, my pee will smell like asparagus. Whereas I know people that can eat like 10 stalks of asparagus and it doesn't smell at all. And there's a few different theories as to why that is. It could be something with smell. Maybe people who say that they don't smell anything after they eat asparagus is because they literally don't have the receptors for that smell. Or maybe it's that different people have different reabsorption rates for asparagic acid. So it could be something in the kidney that has to do with that. Maybe it's something in the nose. Who knows? As always, before I move on to the next topic, let me quickly summarize everything that I just talked about. Blood makes its way into the glomerulus through the afferent arteriole, and some of the components of the blood are filtered into the nephron, and that is known as the filtrate. So that first step is filtration. It occurs at the glomerulus and the Bowmans Capsule. And then once you get into the nephron, you make it to the proximal tubule. And at this step, you reabsorb a lot of the good things that your body wants, like amino acids and glucose. Then you make it into the loop of Henle. And the first portion, which is known as the descending limb, is very permeable to water, but not permeable to ions. So you get reabsorption of a lot of water, and this is due to a difference in osmolarity between the inside and the outside of the nephron. Then you make it to the ascending limb of the loop of Henle, and this is where the nephron becomes impenetrable to water, but ions are let out. So then you get reabsorption of some ions. Then you have the distal convoluted tubule, which you reabsorb some sodium, some chloride. Then you also excrete a few different molecules. And then you get to the collecting duct, which determines the final concentration of the urine, pretty much through the reabsorption of water. And this is stirred on by a hormone. And this is the topic that I will discuss next, is hormones that affect the nephron. All right, so there are 3 key hormones that affect the kidneys and more specifically affect the nephron. The first is antidiuretic hormone, or ADH, and this is sometimes also called vasopressin. The second hormone is called aldosterone, and the third hormone is called natriuretic peptide hormone, which to me is the most interesting of the 3 and also the hardest to say. That literally just took me like 15 different takes to actually say natriuretic right. So it is an atrial natriuretic peptide hormone. All right, so let's get into the first of these hormones, which is antidiuretic hormone, or vasopressin. So antidiuretic hormone is synthesized in the hypothalamus, and it is a peptide hormone. And it's very important for the maintenance of blood tonicity, or blood osmolarity. And what it does is it controls the concentration of solutes in the blood. And so ADH is stored in neurons within the hypothalamus, and these neurons actually have what are called osmoreceptors. And if you think back to physiology, these are just receptors that respond to changes in blood osmolarity. And they're very, very, very sensitive. For example, they can detect as little as a change in 2 milliosmoles per liter. So that's a teeny change in concentration. So you imagine changing from 18 milliosmoles per liter to 20. Well, these really sensitive neurons with these osmoreceptors can sense that. And just an interesting aside, these same osmoreceptors in the hypothalamus are responsible for your feeling of thirst. And so these slight increases in osmolarity that are detected by these neurons in the hypothalamus result in the secretion of antidiuretic hormone. And antidiuretic hormone acts primarily on the collecting duct by increasing the number of aquaporin 2 proteins that are in the cell membrane of cells that make up the collecting duct. So what does that do? That just allows more water to be reabsorbed. So when you secrete this ADH, you get more aquaporins, which means more water can be reabsorbed. And so how exactly does this mechanism act? Well, what happens is, if you think back to what I said before, ADH is a peptide hormone. So what do peptide hormones do? Well, if you haven't listened to my podcast on that, then you might not know. And what they do is they bind extracellularly. So they're going to bind to a receptor on the outside of a cell. In this case, ADH binds to a receptor called the V receptor on cells in the collecting duct. And that is going to trigger a G protein coupled receptor pathway that goes on then to trigger the movement of aquaporin 2 proteins to the membrane of these collecting duct cells. So what causes ADH to be released? Well, anytime you need to increase the plasma fluid volume or decrease the osmolarity, ADH will be stimulated. So if you're reabsorbing just water, that's either going to dilute your blood, which is decreasing the osmolarity, or it's going to increase your plasma fluid volume, or both. So anything that drastically reduces your plasma volume will immediately induce ADH to be released. So imagine you're vomiting, maybe you've had a long night, you're vomiting the next morning, whatever. Your hypothalamus is going to start releasing a lot of this ADH because you're trying to increase your fluid volume. Or maybe you are in Game of Thrones and you get your hand chopped off, shout out Jamie, and now you're losing a lot of blood and therefore you're losing a lot of plasma fluid volume. So your hypothalamus is going to be, again, releasing a lot of this antidiuretic hormone. In addition, it will be stimulated whenever you increase the osmolarity of your blood. You might have eaten a lot of salts, eaten a lot of glucose, whatever it is. This will stimulate your hypothalamus to produce ADH. So what's the takeaway here? Well, ADH or vasopressin increases the reabsorption of water in the collecting ducts by increasing the number of aquaporins that are in the cell membrane. The next hormone I want to talk about is aldosterone. And aldosterone is produced in the adrenal cortex, and it is a steroid hormone, i.e. it is made of this 4-ring structure that's derived from cholesterol. And it plays a big role in sodium and potassium regulation, as well as blood pressure regulation. And it does this by stimulating the reabsorption of water and sodium and increasing the secretion of potassium. So that's to say that it helps you retain water and salt, sodium salt, and it helps you get rid of excess potassium. And to understand aldosterone's mechanism of action, you need to understand what steroid hormones do to act or how they act. And again, you can go back to my hormones podcast, but essentially how they act is they are able to diffuse through the cell membrane and they bind to these nuclear receptors where they then affect transcription. So it's a much slower process than, say, like a peptide hormone, but just know that they go all the way inside the cell and they affect transcription. Still, the most important thing to know, obviously, in terms of renal physiology is where aldosterone acts. So aldosterone acts primarily on cells in the collecting duct and in the distal convoluted tubule. And so essentially what aldosterone does is it will go to these cells in either the distal convoluted tubule or the collecting duct, diffuse through and bind to nuclear receptors. And what that then does is it stimulates the production of these different membrane transporters. So it starts increasing the transcription of the mRNA that then gets transcribed to make these proteins that then act as different channels or different transporters for sodium ions and potassium ions, and also water. All right, so that is aldosterone. Let me quickly recap that. So the takeaway here is that aldosterone acts on the collecting duct and the distal convoluted tubule by increasing the reabsorption of sodium and increasing the secretion of potassium. And so the secretion of more aldosterone will increase blood osmolarity, leading to a higher blood volume, whereas less aldosterone will result in a lower blood osmolarity, also a lower blood volume, and if you were to take a little swig of your urine, it would be more salty. The next hormone is called atrial natriuretic peptide hormone, or ANP. And this is, as I said before, this is a sweet system, super cool. So this hormone is a peptide hormone, and it is actually secreted from cells in the cardiac atria, which is the heart. And this hormone is released in response to overstretching of the heart. And so essentially what happens is when your blood volume is too large, your heart can kind of tell it is being overstretched. It's like, damn, you got a lot of blood here. What the hell's going on? And it responds to that increase in blood volume or that extra stretch in the heart by releasing this atrial natriuretic peptide hormone, or ANP. And so at this point, I hope that you're thinking, okay, well, this hormone is released when the blood volume is too large, then it must act to decrease blood volume. And if you're thinking that, you're right, because that's what it does. And so how does it do this? Well, mechanistically, it is a peptide hormone. So it binds to the exterior of cells. And in this case, it binds to some non-G protein coupled receptor protein. And in this case, like others, this binding triggers an intracellular signaling pathway that eventually affects something within the cell. And you don't really need to know what exactly it affects within the cell. I'd know just kind of the big global things that it does to the nephron and to the kidneys. So it does a bunch of different shit. And so it increases glomerular filtration rate, which is the rate at which fluid is filtered through the glomerulus. And so obviously there you're getting rid of excess fluid. And it also acts to decrease sodium and chloride concentration in the distal convoluted tubule. And so remember, what happens when sodium and chloride are reabsorbed is sometimes water will follow that to try to even out the osmolarity between the nephron and the blood. And so here, that's just another mechanism that helps you excrete essentially more water. And so remember that typically water will kind of follow these ions as it tries to even out the osmolarity between the nephron and the blood. You know, it's a little bit like that one annoying friend who just kind of follows you around a party. You know, you're trying to go talk to different people, be social, and you just got this person that keeps sticking around by you. That's water, essentially, in this case. So again, you decrease the amount of sodium and chloride that are reabsorbed in the distal convoluted tubule. The third thing that this ANP does is it decreases renin production. And renin is just a hormone that is produced in the kidneys that stimulates you to drink more water. And so essentially what's going on there is it is decreasing the signal for you to drink water. And therefore you drink less water and your blood volume doesn't increase because you're drinking more water. The takeaway here is that atrial natriuretic peptide hormone acts in opposition to aldosterone. It decreases plasma volume through a bunch of different mechanisms. All right, so those are the three hormones that you're going to want to know for the MCAT that have some kind of effect on the kidneys and more specifically the nephron. The last thing I want to talk about here are different measurements of renal function, one of which I just mentioned. If you didn't catch it, you will catch it in a second. All right, so there are a few terms to be familiar with in terms of measurements of renal function on the MCAT. The first is glomerular filtration rate, or GFR. And so this is the volume of fluid filtered from the renal glomerular capillaries into Bowmans Capsule per unit time. So this is essentially how much of the plasma is filtered in to the nephron per unit time. That's the glomerular filtration rate. And this is a pretty good indicator of kidney function. So if you have a pretty normal glomerular filtration rate, then you know your kidney's functioning all right. But if you have a really low or decreased GFR, then you know something's probably going on with your kidneys. You know, maybe it's an indicator for kidney disease or renal failure, whatever it may be. Decreased GFR is not good. The next term you may see on the MCAT is clearance. And so this is the rate at which a compound is removed from the blood. It might be a drug, it might be medication, whatever it is. The rate at which it is removed from the blood. And a lot of times this is synonymous with renal clearance, which is just how fast the kidney is at clearing a compound. And renal clearance is a function of a lot of different things, right? It is a function of the glomerular filtration rate. How fast is this substance being filtered into the nephron? It's also a function of reabsorption in the different parts of the nephron. So, you know, how easily is this compound reabsorbed back into the body? It's going to be cleared slower if your body reabsorbs a bunch of that. And it is also a function of secretion into the proximal tubule. So if a compound is easily secreted into the nephron, it's going to be cleared quite a bit faster. The next term you might see is renal plasma flow, which is the volume of blood plasma delivered to the kidneys per unit time. And the last term I want to discuss here is the filtration fraction, which is the glomerular filtration rate over the renal plasma flow rate. And so this is essentially the fraction of plasma that is filtered through Bowmans capsule into the nephron. So for instance, if you have a filtration fraction of 20%, that means that 80% of the plasma is left in the blood and then follows up through the efferent arteriole and into the pertubulary capillaries. And I wouldn't worry too much about trying to understand changes in filtration fraction and renal plasma flow and, what that indicates in terms of the health of your kidneys. What I really would understand is, number one, the terms themselves, understand what they mean, but mostly understand that when you have a decreased glomerular filtration rate, that is bad, and that is an indicator of some kind of kidney problem. All right, that is it for the Renal Physiology podcast. Thanks for listening, and as always, if you like the podcast, please go leave us a review on iTunes. It helps with our ratings and helps us reach more and more students. Thanks again. Hope you come back for the next one. All right, time for a segment that I call MCAT Advice of the Day. And this is kind of MCAT advice, kind of not. It's more like med school advice, but it's a little bit related. So I was talking to one of my friends the other day, and I think she brought up a really good point. She was saying that she was talking to some college students, and they're basically saying that if they didn't get into med school straight out of college, it was over for them, and they would be pretty upset if that didn't happen. And I think it's important to point out that I think on average, people that make it into med school tend to take like 2 or 3 years of gap years in between college and med school. And I think a gap year or two can actually be a really, really helpful, important thing. One, for building your application, and two, you have your whole life to be a doctor if that's what you really want to do. So my advice is this. If you're kind of teetering on the edge right now and saying, well, I really want to take the MCAT now so that way I can get into med school straight out of college, or maybe you just took the MCAT and you're trying to prepare to apply this cycle, but you didn't do so well. And so now you're kind of like questioning, what do I do? Should I just try to throw this out and see what happens, or should I take another year? Or you're asking yourself any of these questions. You're just trying to figure out, what's the best way to go about applying to med school? When should I take the MCAT? Any of it. My advice is to take a gap year if you're in that position. If you're really questioning it, take a gap year. Do something that you really, really love to do, whether that's singing in a band, working on some research you're passionate about, maybe get involved with healthcare policy or start some kind of educational initiative. Whatever it may be, do something that, number one, you love doing, but that will also help you build your med school resume and make you a very unique applicant. I'm in the process of applying right now, and I've talked to quite a few different, students, med school students, or doctors that are now serving on the board of admissions for different schools that have really said, we're looking for unique students, and, what will set you apart from these students that are all applying. So I think that's a huge thing, is being able to say, I've done this for a year and I don't think anyone else has, or to be able to go to an interview and be super excited about this thing you've done for a year or two, and kind of impress those admissions committees. And of course, there's nothing wrong with going straight from college into med school. I know people who have done it. It's not a bad thing to do. All I'm trying to say is if you're teetering on the edge here of thinking, okay, should I rush the MCAT? Should I not? Just know that there's nothing wrong with taking a few years, doing something completely different, hopefully, obviously, still in the field of medicine or education or something that's going to help you get into med school. But there's nothing wrong with that. So just keep that in mind as you start figuring out when you want to take the MCAT, and and maybe you have to take a retake and you're really upset because it's set you back a year or two or whatever. Don't be that upset. Be like, okay, this is my opportunity to do something unique and make me a better candidate when I do apply to med school.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com for the experts.